So that was a pity, to say the least. But we decided to look in detail. We had to go back to the drawing board, which is, of course, what we do. And we had to go back and look at why this is. Now, if you look at the structure of BBr346, where it's trinuclear, it's linked by these diamines. The overall charge is 4+, and we always see pre-association to biomolecules. We've done extensive NMR studies on DNA with 15 nitrogen. You always see a pre-association. We've seen it with human serum albumin and other proteins. You also have a pre-association in the pharmacokinetics in plasma, which we believe to be electrostatic. So we decided to make, instead of making a covalent compound, make a totally non-covalent compound. We said, well, why don't we look at the electrostatic interactions and the hydrogen bonding interactions in the absence of a platinum chloride bond? So we made both an ammonia here and a dangling amine. This is the most important compound. And so now we have no substitutions possible. We only have charge and hydrogen bonding. So we can examine non-covalent interactions with inert platinum amines. So we did that, and we were able, because of the charge, we thought, well, maybe we'll get some crystal structures on DNA. In fact, we noticed this: if you take these highly charged species and add DNA, even in a test tube, do UV visible or a melting point, if you're not careful, the thing just precipitates out because of the neutralization of the charge. So we decided to try to crystallize these compounds. This is the Dickerson-Drew dodecamer. This is a 12-mer duplex, and this was the first small piece of DNA to be crystallized as a single crystal and shown to have the double helical structure. Okay, so we got very nice crystals of both the 6+ compound and also the 8+ compound, and it gave quite an unusual result. They're only hydrogen bonding interactions on DNA. This is the crystallography, and you will see here that this is the platinum molecule. Here's an ammonia, and here's an NH2 linking the other one, and it forms these small rings with the oxygen of the phosphate. We call it a phosphate clamp, and then the distance between the two platinums is long enough that we can span the minor groove to form a phosphate clamp on the other side of the DNA and on the other strand, or in fact we can move down the strand and we can span, we can backbone track. We can track the backbone of DNA with one platinum here, a second one here to the next phosphate, and a third one to this phosphate here. So that's the way these compounds bind. And every single motif is like this. There are 17 contacts between DNA and this trinuclear molecule, 18 contacts. 17 of them are this modular unit. We have an ammonia, platinum, NH2, oxygen. And it was pointed out actually by a referee that this is very similar to how arginine recognizes DNA. This is a model for arginine with the amidine groups binding to phosphate. And the similarity would be that this has to be rigid. The reason why arginine is rigid is that it's delocalized over the 2 CM bonds. The reason why this is rigid is you have to maintain 90 degrees around the platinum square plane. So although the compounds may look like a polyamine, polyamines are very flexible and they don't have this rigidity. So we've looked at this compound now in considerable detail. Neutralization bends DNA about the same way as cisplatinum, and we believe that this binding is very distinct. So it's actually the third mode of DNA binding recognized after intercalation and minor groove binding. Here, for example, is another comparison with the arginine and the phosphate, and this is a typical example of arginine binding to DNA. This is the tata binding protein TBP binding to its cognate DNA, and here's an arginine, an arginine, and an arginine. This is how TBP recognizes DNA. TBP is a protein involved in initiation of transcription. And I won't have time to go into detail, but in actual fact, our compounds are strong enough that they will prevent TBP binding to its cognate DNA. Okay, so If you look at intercalation, of course, as you saw in the previous talk with some of the ruthenium DPPZ compounds, require these delocalized, at least three-membered rings to intercalate, stack between the bases, and extend the distance between the bases from 3.4 to about 6.8 angstroms. So clearly this is a different mode of binding compared to the phosphate clamp. More interesting is the minor groove situation, because we've done NMR on these compounds and we see contacts in the minor groove. And we just switch now. Here is a model of Herx dye, a classic minor groove binder, and this binds in the minor groove through hydrogen bonding interactions of these NH groups with oxygens of thymine. Here's an oxygen here of a thymine, here's an oxygen of the backbone, and the minor groove binder lies across the groove. And here is our typical phosphate clamp. Here's the oxygen of phosphate. Here's one platinum with the two ammonias. Here's the second one, and then here's the third one down here. It's a bit obscure. So we think that this is a distinct mode of binding with consequences for biological activity. So now I'm going to switch. We had done that, and this was interesting. But one of the things that we had noticed over the years is that these highly charged compounds actually got into cells better and quicker than cisplatin. And this was, again, counter to what we had expected, because the original idea for cisplatin was that it was a neutral molecule that entered cells passively and then was hydrolyzed to bind to biomolecules. But if you look at accumulation, we see accumulation increases with charge. Here's 4 plus for the BBR346 for trinuclear drug covalent. Here's 6 plus, and here's 8 plus. And these are inside cells. We did all the appropriate experiments to indicate that we're not looking at platinum sticking to the membrane, although I'll show that in a moment. Cisplatin would be just on the axis here. So we see more platinum getting into cells. And this has a consequence, because this compound binds to DNA but not covalently, but we get a lot of it into cells. It's not decomposed by glutathione, and so in fact we began to see reasonable cytotoxicity for these non-covalent compounds in comparison to cisplatin. So this is unusual. It's 8 plus, it's got no chloride, and yet cytotoxicity is equal to cisplatin. And we've done this now many cell lines, and we see the same result. So for many years we said, well, we know more gets into cells, but we don't know why, but we don't know how cisplatin works either, so we decided not to really spend much time thinking about it. And then finally we had to address the issue. Mainly because it's becoming more and more obvious that accumulation defects are really important in clinical resistance. We've recognized now about 6 different pathways in which cisplatinum can enter cells, or platinum drugs. I'm going to talk about this one because this is unique to our compounds. But if you plot cisplatin accumulation versus a range of cell lines and against fold resistance, you find a reasonable correlation. So this issue of accumulation is becoming as important as tolerance of DNA adducts in terms of how a cell or a patient develops resistance to the drugs. So we liked this idea of the polyarginine analog. So we had a compound that looked like a polyarginine. And polyarginine does a bit of everything. It's many proteins which transport drugs into cells have protein translocation domains which are very rich with arginines and lysines. And so we decided that maybe we should follow the analogy. And if you look at the literature, you find that arginine here, a sulfate, here's a phosphate like a phospholipid. It'll bind to the membrane. But the way polyarginines enter cells is they bind to sulfates of heparan sulfates, the glycans, on the membrane of DNA. I beg your pardon, the membrane of a cell. So this was confirmed. It's recognized that this is the way polyarginines enter cells. And so we thought, well, maybe since sulfate is isostructural with phosphate, maybe this would be a mechanism for entry of our compounds into cells and explain why we have such a high activity. So we started to look at binding of small anions, and just I wanted to point out to the students here that it's interesting. In general, we would say that carbonate and sulfate and phosphate are not good ligands for platinum because they all contain oxygen. But if you're present in plasma at 24 millimolar, then it becomes a good ligand. It's very simple. So you should remember that. So these are weak anions, and they may well be involved in trafficking of platinum compounds. So the experiment you can do is you can buy fluorescent arginine. And this is a nice experiment because fluorescent dyes have been used to attack the cisplatinum, but they change the pharmacokinetics, and they're not really relevant. But in this experiment, you actually put the fluorescent dye, rhodamine or fluorescein, on a polyarginine, on arginine 9. And it gets into cells, and here you see, this is the experiment that we repeated this. This is the experiment that proved that the glycans were receptors because these are CHO cells, and this is a knockout of a heparin sulfate, and this is a knockout of a heparin sulfate and a chondroitin sulfate. And you see in the normal wild-type cells, you see lots of fluorescence inside the cell. You can quantitate this. If you knock out heparin sulfate, you see less. And if you knock out heparin sulfate and chondroitin sulfate, you see even less fluorescence. So this, along with other experiments, really confirmed that these oligosaccharides, glycans, were receptors for polyarginines. So the experiment we can do is we can administer a platinum compound 5 minutes prior to the addition of a fluorescent dye and watch the fluorescence. So here's the control experiment. If you administer cisplatinum, you see the same amount of fluorescence. So cisplatinum is not affecting the entry of the tamra-R9, the polyarginine, into cells. If you do oxaliplatin, the mononuclear drug, the same thing happens. But when you go to our charged compounds, you see a diminishing of fluorescence for the clinical drug, BBR3464. 6+ you see more and more, and 8+, you really almost totally eliminate the binding of tamra-R9 to its receptor. So this is a good argument that we are competing with the polyarginine for the polyarginine receptor, which are these proteoglycans on the cell surface. And you can confirm that this is a mechanism of accumulation for our compounds. You can measure platinum uptake and cytotoxicity, which also matches this. In the wild type, it goes down in the heparin sulfate knockout, and it goes down even further in the heparin sulfate chondroitin sulfate knockout for this 8+ compound. Similarly for our 6+ compounds and BBR3464. And you see also the extra accumulation. This is actually normalized against the for 3 platinums, but even so, you see there's much more platinum in the cell than for cisplatin and oxaliplatin, and you see that cisplatin and oxaliplatin accumulation is unaffected by the nature of these 3 cells. So we have a situation where we have a new mechanism of cellular accumulation through proteoglycan binding, heparin sulfate receptors on the surface of a cell, but also it differentiates the compounds even further from cisplatin and oxaliplatin because they do not they are not affected. The receptors are not receptors for cisplatin or oxaliplatin. This also works in tumor cells, and the reason why this is of interest is because many glycans are overexpressed in many tumors. It's hard to quantitate because the nature of the glycan is very complicated. We've looked extensively at the literature, and it's all over the place. You can't really pick a cell line and say this is a cell line with a lot of heparin sulfate, but certainly it's understood that heparin sulfates are overexpressed in many tumors, and this could be a unique way of obtaining tumor selectivity. The problem with cisplatinum, it works very well when it works, but it's cytotoxic, so it's indiscriminate, and so perhaps this mechanism of accumulation would be a mechanism for tumor selectivity. Here's a colon cell line, and you see this is the relative fluorescence. You can do the same experiment with the tamra-R9, oxaliplatin and cisplatin not really affected. Once you start adding charge, you see that in the tumor cell lines, HCT116, which is colon, or osteosarcoma, you see again a significant decrease in fluorescence, indicating that in tumor cell lines, these compounds are also getting in through the heparin sulfate pathway. And it seems to be a larger difference for the osteosarcoma than for the colon cancers. And actually, our compounds are quite active in osteosarcoma. So we're beginning to piece together pieces of evidence that we had from 20 years ago and never really understood why. And I said, oh, maybe that's the reason. And now we're beginning to understand some of the biology of these compounds. Okay, so I'll skip that. The next question we then have, so we have the situation where we've looked and found a new mechanism of cellular accumulation. But let's think about what would be the consequences of binding strongly to a heparin sulfate on a surface. And how does that affect the biological function of a heparin sulfate and a heparinase? Okay, so what are the functional consequences of platinum binding? Let me just show you first of all. I'll go back to this in a moment. Let me show you just some basic chemistry. You can take an octosaccharide and bind it with mass spectrometry, and you can show very clearly that these compounds form one-to-one adducts. I do not want to be an oligosaccharide chemist. Trust me, it's the last thing in the world I want to be. But it is known that the sulfates are very labile on glycans. And so if you run a mass spec without any MSMS, just the mass spectrum, you will see a range of peaks. It's quite difficult to analyze. You'll see the free octosaccharide, you'll see loss of a sulfate, loss of two sulfates, and you see this envelope of peaks corresponding to labelization of these sulfates. This is a little bit out of focus. If you coat this octosaccharide with a polynuclear complex, reduce the charge, you also stabilize against sulfate loss. So this is showing at 10 electron volts for our 8+ compound. This is free oligosaccharide. You see about the major peak is about 6 sulfates lost. We only see 1, so there's a stabilization of about 5. You increase the energy, you see more loss for the free oligosaccharide, and again stabilization for the platinum compound, and also as you increase the voltage up to 30, you see the same situation. This is a biophysical confirmation that we are actually able to bind these compounds to oligosaccharides, just like we can bind them to DNA, except that the negative anion is now sulfate rather than phosphate. So what are the consequences? Well, we know that heparin sulfates binds growth factors such as vascular endothelial growth factor, VEGF. They're involved in cell-to-cell adhesion as well as cell-to-extracellular matrix adhesion. So heparin sulfates are extremely important in the development of new basement membrane tissue for cells. There are associated enzymes with it. Heparinase with an A is a mammalian enzyme which cleaves heparin sulfate, increases tumor vascularity, and heparinase with the I is a bacterial enzyme which does similar things. And the basic concept is that the enzyme heparinase will come along in tumor cell invasion. It will cleave our heparin sulfates, which are these blue things, and release growth factors into the medium, and cells can invade through basement membranes by release of these growth factors by cleavage of the heparin sulfate, the long oligosaccharide polymer from this enzyme. Again, heparinase is overexpressed in many tumors, and overexpression is correlated with poor post-operative survival in patients. So the oligosaccharide community developed 4 or 5 saccharides, tetro, pentasaccharides, to try to mimic the inhibition, to inhibit the enzyme by peptide mimics to prevent the heparin sulfate cleavage, and some of these are in the clinic.